Wajid from Pune National University. The topic of my today's talk is characterization of bio-cellulose produced by self-free enzyme system. The contents covered will be introduction, methodology, result, and discussion, conclusions. If you look at the biomass composition, it contains 43% by weight cellulose. But this is the most abundant polymer, as we know, as well as it is non-toxic, biodegradable, and biocompatible in nature. However, it encounters several limitations, such as it contains impurities in the form of lignin and hemicellulose. Due to these impurities, it has low mechanical strength, low biocompatibility, and high cost of processing. There are several routes for the synthesis of biocellulose, such as the plants, enzymatic synthesis, and chemical synthesis. However, the most important, attracting topic for today nowadays research is the production of biocellulose through microorganisms. The cellulose produced through microorganisms is called microbial cellulose. As we know that Azotobacterium xylenum and Gluconacidobacter hansenii has been reported for the production of microbial cellulose. The produced cellulose is in the form of a hydrogel. It has same chemical properties, chemical structure as that of plant cellulose. However, compared to plant cellulose, it has some improved properties such as better physical properties because it is pure and containing no hemicellulose, lignin, or some minerals. It has high water holding capacity and slow water release rate. Moreover, it has enhanced mechanical properties such as high tensile strength and crystallinity, and as well as it has better biological properties as biocompatibility toward the animal cells. If you look at the synthetic route of cellulose through microorganism, it is a complex metabolic pathway that involves mainly the cellulose synthesis from glucose through a series of enzymatic reactions. However, besides the main synthetic pathway, there are several interconnected pathways that are connected with the main pathway that results in a complicated metabolic system. Several approaches have been used to improve the cellulose synthesis, such as the conventional fermentation approach, metabolic engineering, genetic engineering strategies, strain improvement methods, mix or co-culture. But nowadays, attention has been given toward the synthesis of cellulose through cell-free system. What are the advantages of cell-free system compared to conventional microbial system? As we can know, in the cell-free system, pH, ionic strength, and cofactors concentration can be easily controlled. Moreover, there is a lower risk of contamination by other microorganisms since no microbial cells are involved in the system. And enzyme concentration can be easily adjusted. Moreover, the enzyme of desired features can be introduced in the metabolic pathways, as well as there is a maximum and uniform enzyme substrate interaction during the reaction. Moreover, the process is less labor intensive that results in improved product yield. The product can be easily recovered and purified, that results in the lower capital cost. If we compare the cell-free system with the wholesale system, in case of wholesale system, we can see that expensive media components are used because beside the main carbon source for production of cellulose, there are several nutrients that need to be added for the growth and proliferation. As a result, the substrate is used for feed and proliferation beside the product formation. This results in lower yield. Moreover, the process is aerobic, and there is a high risk of contamination by other microorganisms. If the production is carried out in agitation culture, then it can result in the cellulose negative strain formation that will result in the lower production. Moreover, there is a limitation in the nanocomposite synthesis as live cells are involved, and the nano materials can easily kill the microbial cells. But in case of cell-free system, the process is cost effective as only carbon source is required for the production of cellulose. It results in high yield. The process is anaerobic because no live cells are present, as well as there is a lower or no risk of contamination. No microbial strain, negative strain formation can take place, and no microbial cells are involved. Moreover, this process can favor the synthesis of nanocomposite with different bactericidal materials. So we have designed a strategy for the cell-free enzyme system for production of biocellulose. The system was developed through the simple bead beating method that results in a complete cell lysis, as we can see no microbial cells when the cells were lysed through bead beating. Moreover, then the obtained cell-free lysate was analyzed for the presence of different enzymes that involved in the complex metabolic system of microbial cells through SDS page analysis and LCMS, MS, LTQ or Bitrop analysis that confirmed the presence of all essential enzymes required for the biocellulose synthesis. In our experiment, we carried out the biocellulose production using the microbial system and cell-free system for 3 days, 5 days, 10 days and 15 days. So we observed that cellulose production takes place in both the system. But when we analyzed the media after completion of cellulose production, we observed that no cells were present in the media in the bio-cellulose system for by cell-free enzyme system. This was further confirmed through SEM images, as we can see that no microbial cells are present shown by the agar plates as well as the SEM images. But in case of microbial system, we can see that colonies appear on the agar plate as well as microbial cells were shown on the SEM images. These results show that in case of cell-free system, bio-cellulose production was solely carried out by the cell-free enzyme and not by the microbial cells. Moreover, the cellulose produced by microbial system and as well as the cell-free system was analyzed through SEM. As we can see that the SEM images show that the cellulose is composed of complex microfibrils, but in case of microbial system, the network is less compact as compared to the cell-free system. That is more compact and the fibrils are tightly arranged. As well, if you see the cross section, the cellulose sheets cellulose is produced in the form of layered structure. The chemical structure of biocellulose and microbial cellulose was confirmed through FTIR analysis. That showed that the chemical structure, as I described earlier, the chemical structure of microbial cellulose and biocellulose is similar. That contain same type of chemical groups. The XRD analysis was carried out to study the polymorphic structure of microbial cellulose and biocellulose. As we can see, the different peak positions were observed in the microbial cellulose as compared to biocellulose. We can see that the biocellulose structure XRD pattern was similar to that of cellulose 2. But in comparison, the microbial cellulose structure was similar to that of cellulose 1 polymorphic structure. So these results were further confirmed through the NMR analysis. We can see that all 6 carbons were present in both type of cellulose, but the position of carbon 6 was shifted toward the lower ppm. That showed the structure of cellulose 2. So both XRD and NMR analysis concluded that the cellulose produced by a cell-free enzyme system was actually cellulose 2 polymorphic structure. Similarly, water holding capacity and water release rate was also analyzed. So we observed that the water holding capacity of microbial cellulose was much higher as compared to the microbial cellulose. Moreover, when the water release rate was analyzed, we observed that a steady decrease in the water release in the case of biocellulose, but in comparison there was a rapid decrease in the water release in case of microbial system. And we observed that after 20 hours, 68% water was still retained in the microbial system. 68% water loss was carried out actually in the biocellulose system, but in comparison 85% water was lost after 20 hours. Mechanical properties of both cellulose were analyzed by analyzing the stress, strain and Young modulus. As we observed, a high stress and Young modulus for biocellulose compared to microbial cellulose, but a lower strain for biocellulose compared to microbial, and this was obvious as it is inversely related to the stress. So in conclusion, a GHNI-PJK based cell-free enzyme system was developed through bead beating. The presence of all essential enzymes was confirmed through SDS as well as LCMS LTQ arbitrop analysis. The system successfully produced biocellulose. The yield of cell-free system was comparatively better as compared to microbial cellulose system. The FESM analysis confirmed the biocellulose produced by cell-free enzyme system was only as well as and has better arrangement of fibers compared to microbial cellulose. The basic cellulose structure was confirmed through FTIR analysis, and the XRD analysis showed that biocellulose has cellulose 2 while the microbial cellulose has cellulose 1 structure, and this was further confirmed through NMR spectroscopy. The biocellulose showed high water holding capacity and slow water release rate compared to microbial system. This was a useful feature for the biomedical application. Moreover, the mechanical properties of biocellulose were comparatively better than the microbial cellulose. Thank you so much. This is my research group.